Okay, so today we will be looking at Watson and Crick's DNA structure. DNA is double-stranded, forming a double helix. The strands are also anti-parallel and have a platonic coil that can't be pulled apart. Looking further into that, I've drawn an unwanted DNA, and you can see the different nitrogen bases pairing up. So what makes up DNA are your nucleic acid components. The first is deoxyribose, and I actually forgot to label the carbons, but the one furthest right is 1' and the one below it is 2', and it keeps going after that. Deoxyribose and ribose differ because on the second prime carbon, ribose has an oxygen, whereas deoxyribose doesn't, hence the name deoxy. Then you have your nitrogenous bases pyrimidines, which are C and T, and purines A and G. Lastly, you have a phosphate group. Here I've drawn the parts put together, making a nucleotide. 1' carbon attaches to the nitrogenous base. The second prime distinguishes between DNA and RNA. The 5' has a phosphate group, and the 3' bonds to the next phosphate group. And here I'm drawing multiple nucleotides paired together. The strands are anti-parallel, so one strand goes from 5' to 3', whereas the other goes 3 to 5 when you look at it from top to bottom. Nitrogenous bases pair together using relatively weak hydrogen bonds, and A pairs with T using 2 hydrogen bonds, whereas G and C pairs with 3. Those are the red dots I've drawn to represent a bond. The bases are hydrophobic in the center, while the phosphodiester backbone is hydrophilic on the outside. The hydrophobic interactions are maximized by base stacking and are the main force in stabilizing the double helix. The phosphodiester backbones are closer together on one side of the helix than the other. Major grooves are when the backbones are further apart, whereas the minor grooves are when they're closer together. When I draw the lines, you can see the difference in the distance between the two. The bottom one is further apart compared to the one on top of it. And this is important because proteins that bind to DNA typically interact with bases on the major groove side because the backbones are not in the way. And handedness refers to the direction the helix is twisting, so it could either be left or right-handed. The way I'm able to tell is by looking at the front helix. If the helix is pointing up towards the right, it is right-handed, and if the helix is pointing up towards the left, it is left-handed. Here I have several left-handed DNA. The orientation of the picture is weird, but if you were to look at the DNA where it is standing straight, you can see that the front helix is pointing up to the left.